my name is Dr. Monstrosity, and today I'll be filling in for Akshaya because sadly she could not attend today. But anywho, today we'll be talking to you about nucleic acids and everything you need to know. So let's get into it. Now, before we start, we need to define some key definitions and terminologies. To understand what a nucleic acid really is, we need to know what a monomer, a polymer, a function group is, as well as what a nucleotide is. So, a monomer is a smaller molecule, whereas a polymer is made up of these smaller molecules and is a big molecule. And functional groups, you might remember periodic families from the periodic table in grade 9, 10, and 11 chemistry and biology. But what a functional group is really similar to that actually. It determines the characteristics and chemical reactivity of molecules. And so why do I need to know this? Cut to the chase. So you need to know this because monomers and polymers are nothing but nucleotides. And nucleotides is what makes up nucleic acids. Isn't that so cool? That's mind-boggling and blown. So let me cut to the chase and tell you what a nucleic acid really is. Well, a nucleic acid stores and transmits the most important information in our body. It's in charge of coding information for the protein structures, our looks, and our susceptibility to diseases. True. God bless me. Anyway, like I said before, nucleic acids are made up of nucleotides. But what are nucleic acids really composed of? Nucleic acids are composed of phosphate, hydroxy, and carbonyl functional groups. But if that's not cool enough, let me blow your mind even more. Here we have types of nucleic acids. There are three types that we're talking about today. We have DNA, RNA, and ATP. DNA is deoxyribose nucleic acid. And you remember from grade 11, the reason we have brown, blonde, blue hair, there's more tall, short, and in between. The reason for that is because we have DNA. We have genetics that are passed down that make us who we are. But now that you already know what DNA is, let's tell you a little bit about nucleic acids and DNA. Well, for DNA, it cannot leave the nucleus. You might be thinking, why not? Well, DNA and RNA are two different things. DNA stays within the nucleus, whereas RNA can leave the nucleus. DNA has a double nucleotide chain, meaning a double helix structure, the staircase-like thing, where instead RNA has a single nucleotide chain. And DNA is composed of deoxyribose sugars in the sugar phosphate backbone, while RNA is composed of ribose sugars in the sugar phosphate backbone. Another cool fun fact about DNA and RNA, the last difference, is the nucleobases. Both DNA and RNA share nucleobases iodine, cytosine, and guanine. But for DNA, you have thymine, and for RNA, you have uracil. And before I finish off with DNA and RNA, one last thing to mention is that there are three other types of RNA that I want to talk to you about. There's messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. Now we're left with ATP, meaning adenosine triphosphate. Adenosine triphosphate is a molecule composed of nitrogenous base A, meaning iodine, with a ribose sugar, meaning the same sugar that RNA is composed of. But why am I telling you this? You understand the importance and the critical roles that nucleic acid plays in our body. So here's the rundown. DNA is in charge of permanently storing genetic information. RNA is involved in protein synthesis. And now for types of RNA, mRNA produces instructions to make proteins fight diseases. We have tRNA, which is responsible for decoding those instructions and turning them into a protein. And then we have RNA, which takes these proteins and make them into ribosomes. And if that's not cool enough, let me explain to you ATP molecules and what their importance and role is. So ATP molecules, also adenosine triphosphate molecules in the body, is that they're the building blocks for storing and transferring energy in cells. But if that's not cool enough, let's tell you and give you a closer look. Here are diagrams. I know we hate them, but we need to know them to get a better understanding. This is how you differentiate an RNA, an ATP, and a DNA diagram. Here, you know RNA is single-stranded, so there's only one sugar phosphate backbone, and the base pairs don't pair like they do with DNA here. Here, DNA is double-stranded, so you have two sugar phosphate bonds, and the sugars in RNA are ribose, and the sugar in DNA is deoxyribose. Now, the difference between deoxyribose and ribose is that there's one less oxygen, meaning deoxy, one less oxygen, chemical prefixes. Another chemical prefix I want to talk to you about is phosphates, triphosphates. How many phosphates do you think are in iodine triphosphate?
Well, if it's iodine triphosphate, I believe that the tri means 3.
Yay, you're correct. There's 3 phosphates because in grade 11, grade 9, and grade 10, we learn about chemical prefixes. But how do I know this is an ATP molecule? An ATP molecule has 3 phosphate groups connected to a ribose sugar, then connected to an iodine molecule. That's an indicator that you have an ATP molecule. But since you got a closer look, let's zoom out a bit and talk about bond structures. Bond structures are really important because when I tell you about the base pairs, apple on tree, car in a garage, the importance of this is because purines have a double ring nitrogen base, where pyramines have a single ring nitrogen base. Purines are iodine and guanine, meaning apple and garage, where pyramines are single ring, so that's cytosine, thymine, and uracil, meaning car, tree, and uracil for RNA, thymine for DNA. After you talk about bonds, let me tell you something to remember this a little bit. Purines are not pure, but they're a giant iodine, guanine, and pyramines are small and cute. And so you have cytosine, thymine, uracil, like cute. Now, another important bond you need to know is a phosphodiester bond. A phosphodiester bond is the actual motion of linkage to a phosphate group plus a ribose or deoxyribose sugar linking to a base pair. And what this does is that it creates a single strand or double-stranded structure of RNA or DNA, which is crucial. And one last bond I want to talk to you about is a hydrogen bond. A hydrogen bond, we already learned this previously in grade 11, but it's just so for DNA, our base pairs can become more balanced and can react with each other properly. Like in this diagram right here, the base pairs are being bonded with a hydrogen bond in between. Now, last but not least, because you guys are so amazing, we have directionality. Directionality, you have to switch up directions and think about it from another point of perspective. For us, DNA sugar phosphate backbones actually run anti-parallel, meaning the end base is point toward the interior of the molecule. And you're thinking, why? Why did I do this to you? Well, you need to know this because that is what ensures base pairing happens. That's what makes sure that A goes with T and G goes with C and etc. But because that base pairing takes place, we have a balance in DNA, RNA, ATP, and nucleic acids themselves. Why is balance so important though? Well, it's important because we have this thing called Chargaff's law. And what Chargaff's law is, especially with DNA, there's always an equal balance of quantity in between a base pair, so a 1-to-1 ratio. If you have 1A, you have 1T. If you have 1G, you have 1C. And the importance of this is so we can determine the strength and showcase the presence of a base. In order to turn this example into its answer, a complementary base pair, all you have to do is take the letter and give it its opposite base pair. So for an A, a T, for T, an A. And if you're doing an RNA, taking this complementary base and turning it into an RNA, all you have to do is take an A and turn it into U for uracil instead. Same with percent. If you're not given a full percentage quantity, you always assume that it's out of 100. So if A has 10, that means T automatically has 10% as well. That means it's 20% out of 100%, meaning 80% is left over. We divide that 80% by 2 and get 40 and 40, meaning 40% for cytosine and 40% for guanine. Now, if you have any questions, I can take them now.
I have a question. For example, if, for example, A is paired with T in DNA, and you gotta change an DNA strand to a RNA strand, what would the A pair with?
The A would pair with the U because in RNA there's no thymine. So when you're transferring A to a T, it would be an A to a U instead.
Thanks.
No worries. Also, like I mentioned beginning of this presentation, before the recording started, I did link the activity sheet that you should have been following with along with complementary questions that you could use to make your own notes. I linked my presentation slides and a copy of notes I made just to look at bond structures and ATP, DNA, and RNA molecules. Thank you so much for listening. I hope you have an amazing day.